are you?
I'm fine. How about
I'm doing fine. We had a time change here, so this is the time of year when everyone gets jumbled up.
you? Yeah, right.
Yeah. So yeah, that's great. Did you have any questions about? You've been interested in GSoC applications, the application period.
No, like, do we have to like submit the applications from April 9th or something, right?
Yeah. So submit the applications by April 9th. They open, I don't know when, and sometime. Well, I think maybe they
Its
open.
opening is
Yeah.
April 7th or 9th, and
Yeah.
ending is like 19th of some
Yeah.
April.
I I don't know what the timing is on the. I always get that confused. Like the applications open, then they close, but you should be working on it. All along. And like I said, I
Yeah, yeah.
can help people with their applications if they send them to me in a reasonable state, and then they get I'll give them feedback.
Okay,
Um,
okay. Yeah, I'm like about to start writing my proposal.
okay, yeah. Sure. Why don't I go over that process? I'll put it in the meeting and then see if I'll point people to it later. So let me share my screen here. All right. And so you can see my board here. So every year, we have applications, and they don't really change. This year, we have the longer projects and the shorter projects. So this is a shorter project, so it's going to last through the summer from, I think, like May to late August. So the first thing you want to have is you want to have a section that's sort of an introduction about yourself and why you're interested in the project. Then you have a short section here where you have sort of a proof of concept. And the proof of concept might be like the model you would use, going through that, a review of the model. So it would be like a review, proof of concept. And it would, like, if you can get your hands on some data, I know I shared some data with people, but I can share more. If you can do like a preliminary analysis, that would be great to show that it works. In past years, people have they've done a short analysis to show how their technique works on real data. So you might do like a little. I mean, in some of the projects in the past, they've done some computer vision. They've shown some examples of how this algorithm was segmenting out the cells and everything. So that'll go on for maybe about a page and a half. I don't know. I mean, they have guidelines on the site. So there's a sort of this intro where you set up your proposal. There's a proof of concept review, which might go 1.5 to maybe 2 pages.
Okay.
And then there's a part here that's very important. And that is this is the schedule. So this is almost the most important part of the proposal. And this is where you lay out how you're going to do things, how you're going to get them done, and in what time frame. So you start off with week 1, and you go down to week, I don't know what the, 12 or 13. I don't know how many weeks they break it into. But for every week, you want to have like a list of what you're going to do. And then, you don't want to start off like saying, I want to learn about graph neural networks, say. That's not really going to work. What you want to do is you want to say, I'm going to do that before the period begins. Then I'm going to start with some reasonable goals. So I want to maybe start off with a little, maybe I've shown in this part here that I've done a little bit of preliminary work. Then I'm going to start ramping up to a bigger data set in like week 3, and then going through to week 4, week 5. I'm going to implement these parts. I'm going to get it working. And then, by the end, you should have the full project at least some version of it running as something that you can execute as a program, because at the end of the program they want you to submit an executable file that runs. So they're going to, at the end of the program, to evaluate you, they're going to take the program, open it up, and run it. And if it runs, then then you're okay, you're passed. So that's the way they want it. And so we have to spend from about week 2 or 3 to the end of the program really kind of getting everything in place to get to that end goal. And so, that could be, there might be some obstacles in your way. You might have things that don't work out the way you want them to. So you want to have in this schedule, I always stress this every year, you want it to be flexible enough so that you can maybe find other things that you can do while you're waiting for something. I remember one year we had, someone had an issue with their ability to find a powerful enough computer to do what they wanted to do. And as they searched for that, they were able to do some other things to put them in place. Sometimes people propose starting off with just getting everything in place and then in the second part of the program optimizing it. Or you might walk through it where, you get data, you analyze some data, you go back and get more data, you analyze that. Depends on the algorithm you're trying to use and what gets you to that end goal fastest, or what gets you to that end goal maybe without so many obstacles. But keep in mind you'll still have obstacles and you'll want to plan for that. This part of the program is interesting because it's something they call the community period. So the community period is a period of 2 weeks at the beginning of the program where you get to know the broader community. So in our case, that's the OpenWorm community. And you're going to be able to reach out to people in the OpenWorm community. I usually expect that people engage in our Slack, maybe some of the other groups. We have some wonderful groups in OpenWorm. We have a movement group, so people who do see elegans movement, and that actually has a lot of machine learning in it, and they're doing a lot of stuff with vision, computer vision. And so, that's somewhere you might reach out and ask if they have some interesting data that you might use, maybe not for the main project, but if it's something, that might help you with your validation, it might be worthwhile reaching out. There's a movement database, which has been sporadically online, so that's something that you can use. There are other groups in the OpenWorm Foundation. There's C302, where they do simulations of neurons. They do other types of simulations. There's Geppetto and some of the other programs. These are things that during the community period we would go over, and you would be able to reach out to these people anytime, but that's the time when we want to get you familiar with the larger community. So I think that's a valuable thing. You're going to put that in your schedule. It'll say community period, and then from week 3 on, you'll be doing your project in steps. You'll be proposing, how you're going to implement algorithms, how you're going to put together, some of your resources to make sure you have everything in place. I know in past years people have used the Colab notebooks as a way to get around some of their computational limitations, because the Colab notebooks allow you to, they give you free computational resources through, I think, Google's Cloud Hub. So that's a resource I don't think a lot of people think about. But I've had 1 or 2 students who have had problems with that in the past, getting a good, they have their own computer and Google gives you money for a computer, but you can also, with a lot of the machine learning stuff, a lot of the deep learning projects, it's, sometimes it's a struggle to find the right resources because you're always running up against the limits of them. So, let's see what else. So that's it. I mean, I wanted to go through that structure of the proposal because I wanted to, every year I get questions about it. And I try to stress that every year. And then I get people, I always accept people sending me a draft proposal. You want to make sure it's not just a loose idea. You want to kind of write it up as a short, not a pre-proposal, but just kind of an outline with some things in it. Maybe try to run your algorithm beforehand and see if it's a viable idea, and then I can give you feedback on it. I can tell you whether this is good, what you need to change. Yeah. So, are there any questions on that?
No, I've understood the format in which the proposal has to be made, and yeah, I'll get back to you if I have any doubts while making the proposal.
Okay. Yeah. And this goes for both proposals. So if people are doing the Axolotl project or they're doing the Graph Neural Networks project, it's the same thing. To kind of go through and show a sort of a proof of concept, then have a schedule. And then at the end, I think you also have to talk about yourself a little bit. But that's something, it's like a CV. I think you have to give you have to upload your credentials or something, but there's a small space for that as well.
Okay.
Well, Susan, how are you?
There we go.
Yeah.
How am I? I'm fine. Dr. Sharif just dumped my project on my head.
In a bad way, or?
No, it's the way he works.
Oh.
So I have to have my whole thing done by May 27th.
Okay.
Yeah.
Yeah.
So he says just hack it. He wants me to grab whatever I can in the way of pre-programmed stuff, and then just throw it into the program and get it to work.
Oh, okay.
So, yeah.
Yeah.
I do have a new stand for the microscope, and worst comes to worst, I could always get a peppercorn or something like that and image it instead of the salamander egg, because I do have some salamander egg stuff, and what I did before was put markings on a peppercorn, and then so they're different colors on different sides. That's harder to do than you think.
Yeah.
But I could try something like that too, so you could do both, because peppercorn's around about 3 millimeters. So find a small peppercorn, do that, and then it might have some guideposts because you've made some markings on it.
Yeah. Yeah, that sounds
so
great.
Would that help?
Yeah, I think so. Isn't that what you did in that paper that you did? You did the biosystems paper where you had the different samples.
Okay. Okay, Harikrishna, did you get a copy of my paper?
Which one in which you?
A ball microscope.
Yeah. Yeah, it was mentioned in the Neurostars website, the second paper.
Okay. Do you have a copy of it?
Yeah, I have it.
Okay. All right, good. Well, there's a peppercorn in there, and I could try to get some more images of it or something similar.
Okay. Yeah, I wanted like a time series data so that I can test it out.
Yeah, well, with a peppercorn, it wouldn't be time series, but at least it would be, you could get a whole image then, or all 10 images of the sides of it. And I'm still hoping that my salamanders might decide to lay eggs sometime.
Yeah.
I don't know. The other person who has eggs is a very busy pre-vet student, and I didn't want to bother her until she's done her term, which is in April.
Yeah.
So I might ask her to do something strange with her salamanders after that, but yeah.
Yeah.
Okay.
Yeah. Yeah. No problem. Hello,
All
Karan.
right. Well,
Yeah.
I'll get some peppercorn pictures, and then we'll try to persuade salamanders in April or May.
Okay.
Well,
Yeah.
end of April. They don't like to lay in any later than June. They won't lay eggs.
Oh.
They just won't. Yeah.
Yeah. Hello, Ishan and Karan.
Hi,
Hi.
Bradley. I'm joining after 2 weeks.
Yeah.
Just exams and all. Can't help it.
So yeah, you guys missed that I just went over some of the GSoC, how to write a proposal. And why don't I go over that really quickly again? So I was explaining that the way I usually like to have the way proposals have been written in the past that have been successful is that you've had proposals where you have like a page of introduction to your problem. So, if you're using a technique, or if you're like, the project, what is it about the project why is it important? And this is just to show that the student understands the why they're doing this. Then there's about a page and a half or two, or maybe even more than that, of proof of concept or review. So this is where you go through your procedure that you're proposing, and you explain it, and then you maybe do some samples. So you get some images, you run them through your algorithm, and show that at least it works in principle. So you might show some pictures of cells being segmented, or something that you're doing, maybe some diagrams to show how the algorithm works, things like that. And then the most important part is the schedule. So weeks 1 and 2 are this community period where you talk to people, say in the Open Room Foundation, which is where we're based, and you can talk to people in different projects. You can get an idea of what they're doing. There are other groups such as the Movement Validation Group, where they're looking at images of C. elegans, adult C. elegans that are crawling around in a dish, and they're trying to segment those worms and trying to figure out the movement poses. So that's something that might be useful to you in a project, sort of as a validation step, or it might be interesting for future work. So that's the community period, and then from week 3 on, you have your work period, which is where you actually implement your work. And you want to be careful because sometimes you'll have things that you'll run into obstacles. And so you want to have some sort of plan to get around those obstacles, to do other things while you're trying to solve those obstacles and so forth. So the schedule is very important to work out and to make it, appear that you're going to be able to do this without too much problem. At the end of the period, I think it's at the end of August, you're going to submit your project, and you're going to have to submit an executable, and that executable has to run in order for you to pass. So that's your end goal, is an executable program. And so this entire period has to be geared towards making that work. So that's the flow. And like I said before, I am happy to review people's proposals as they write them up, get it into decent shape, have a nice flow of your proposal, and then I can give you feedback and to make sure that you're on the right track. Any questions about that?
No, Bradley, that makes sense actually.
Yeah.
There should be a proper, you know, format of any proposal. So I'll just draft my work up by this end of this near the weekend, and I'll send a draft proposal up to you.
Yeah. Yeah, that sounds good. So yeah, I don't know what the timeline is. I think the proposal's open. I don't know when you'll have to look on the website.
It's about April 14th. So there's about a month from now.
Okay.
So that's the deadline for the proposals.
Okay. That sounds good. Yeah. And so that's, yeah. And then, of course, you have to register with the website, the Google's Summer of Code website. Then we're also on Neurostars, which is this site, neurostars.org. Some of you have been here. Some people haven't, but because there are multiple ways to get involved in the, to sort of get into the project. But if you're not familiar with the Neurostars forum, this is where we post the projects. We're under INCF, and we're project series 22. So GNN's is 22.1, and Axolotl is 22.2. So we have a description of the project, some links to some resources here, and then some skills and requirements. And then we've had some questions about the project in the chat here. I mean, you're also encouraged to join our Slack, where we also talk about the project a bit, but the Neurostars is another good place to go for this. So the Axolotl project is, can't seem to find that one. Oh, here it is. The digital microsphere, which is what the Axolotl project is. And here, these papers that Susan was talking about, this paper, I think here, the 2018 paper. I think Susan had a 2020 paper, or 2021 paper, if I'm not mistaken, as well. We might, I don't know if I can add that. I think I can add it. Yeah. So maybe I can get that up there. And it's just the citation. So, but we can, this is the place to be if you want to learn more about the project. I don't think I've had any comments on it. So we'll try to go through Neurostars, and we'll try to go through the Slack for people. Okay, so that's, I think, enough for GSoC for today. Why don't we go on? I wanted to ask, does anyone have anything they'd like to present today? Okay.
I certainly don't. I've got a huge project.
Yeah. Okay, well, again, you're welcome. If you have a demo or something you want to show, you're always welcome to do so. So why don't I share my screen? I have some more things on spatial transcription and protein atlases and things like that. So one of the things I found this week was, and last week I think I talked about this, but I talked about it in terms of just like heat maps, which are these maps where you have like a list of sites or allelic sites, and then a list of like samples. So you have like a tissue sample, you get the DNA out of it, or the RNA or the protein, and you do some sort of chemical analysis, and then you plot out like, expression level or some sequence, and then you can build a heat map from like intensity of expression. You can build a heat map out of like quantitative expression, and then you can also have a sequence analysis that aligns it to certain sites. So you can build those types of heat maps. And so those are useful for looking across species or across samples and seeing how those things vary. Another thing you can do is have like some sort of atlas or some sort of like spatial. We have spatial transcriptional analysis, which is where you try to link gene expression or some transcriptional profile to certain locations in an organism or in an embryo. So in this paper, this is a 4D single-cell protein atlas of transcription factors delineate spatiotemporal patterning during embryogenesis. So this is, I think, in C. elegans where they do this. So yeah, this is done in C. elegans embryogenesis. They use protein fusion fluorescent reporters and four-dimensional live imaging. So in this case, they're using fluorescent imaging. So actually what I explained to you before is one way to do this. Another way to do this is to take a microscope and use fluorescent proteins, which you have these fluorescent elements that are usually some protein from like a marine invertebrate that they use, and they're different, like they fluoresce in different frequencies of light. So GFP is green fluorescent protein. It fluoresces as green when it's hit by ultraviolet light. There's red fluorescent protein. There are other smaller variants of it. But basically, when you have it under this UV light, you can see the image and you can see this fluorescence. And so the fluorescence tells you at different intensities where the expression of these things are, because those fluorescent proteins are in a construct that binds to the thing you're trying to target. So if you're looking at a specific protein, in this case in the cell, that construct will bind to those proteins, and then that element will fluoresce, and it'll give you a signal. So you can actually see it under the microscope. It'll be in one place and not another place, or it'll be at a certain intensity in one place and higher intensity in another place. And then it's a matter of image processing to sort of figure out what the intensities are. So you know, this is a nice method for, you don't have to destroy the sample. You have to, well, you kind of have to destroy the sample, but you don't have to, you get like spatial information. You can have it under a microscope. You don't have to map it to anything else. So it's all in one data set, which is nice. This is an atlas they're building. So they're building this atlas at single cell resolution. So they're able to look at each cell and how these proteins are expressed in each cell. And then from there, they can build an atlas of each cell and its properties within the cell. So let's see if I can find some images. So here's a developmental protein expression atlas. This will give me a bigger picture here. Okay, so this is where they had, they identified 609 transcription factors in the C. elegans genome. 290 transcription factors are selected as targets. So of all the transcription factors that they identified here in this first part of the screen, they screened it for these targets. And then they found 291 what they call protein fusion reporters, and they bound them to 266 of these transcription factors. So this is they usually do a pipeline. They usually do like an initial survey of what they want to use as their sort of target. Then they do some screens to see if they're viable, and then they build the construct and they put it into the cells. And then they're able to get these kind of images where you have this entire embryo. This is a C. elegans embryo, starting from very few cells and then increasing in the number of cells to this stage where you have this folded. This is sort of like where they call this the comma stage, where this ball of cells starts to fold, and then you get this where there's folding over, and this is going to form the worm. I don't know which end is anterior or posterior. I think this is the anterior end or the head, and this is the posterior end or the tail, and it'll bend around a couple times by the time the egg hatches. But you can see that you have that fluorescent protein being expressed. And you can see at this scale, you can see the cells that are distinct here. And then you get some other signals that come up later, some of these green signals. And so you get this, there are problems with things like autofluorescence and noise, but it's a nice method for sort of getting at this in what we maybe call real time or real space. And so this is how they do this. They can trace lineages using this method. They can have cells that have the fluorescent protein expressed in it, and then as that cell divides, its daughter cells will carry on that signal. And so you can trace out lineages. You just get that reporter into a mother cell down here, or what they call sometimes a founder cell, which is at the root of that lineage tree. And then you can follow it through to the daughter cells. So you can go from like a single cell expressing this to 256 cells expressing this. And it just shows you that lineage expanding. And then this is their protein fusion reporter for protein expression quantification. So they're actually using two different types of markers here. They're using this cherry red for tracing the lineage, and they're using this green for actually getting the level of protein expression that they're interested in. So in this case, they're looking at the cells, they're tracking the cells, and then you start to see these green spots, and that's where you're getting the protein of interest that's coming up. So you actually have, all you have to do in the experimental setup is switch between the different filters, between the cherry filter and the green filter. And you can get both sets of images in the same sample. And then, this shows you this tracing the protein expression out in a cell lineage. So when you do this kind of thing where you label it twice for different purposes, you can actually build something like this, which is a lineage tree. You have these, you have the green branches, which are where that protein is being expressed. You have the branches that don't have a color, which are branches that exist, but they don't have that expressed protein in it. And you can see then at the end, in C. elegans, it's a nice system because it's deterministic. These cells are going to become, we know what their fate is going to be when they're interadult, when they terminally differentiate. So we know that these cells are going to become neurons, these cells are going to become pharynx, and so forth. And so we don't have to guess, we don't have to do any sort of going back to the adult and checking, because they're all basically going to become that kind of cell. In, like humans or mice, there's a lot of signaling that determines what the fate of a cell is going to be. So it's different in that way. So C. elegans is a nice system for this because you can trace out the lineage, trace out the protein expression, and then know what cell it's going to be. And then that's actually confirmatory in a lot of ways. And then this is your another example of the atlas where you have the cells, sort of, this is where they're in their spatial locations. This is where they're expressing different things. You have these different colors that they've put on here. And then for these different factors, transcription factors. And then this is a lineage tree where you can see these things here being expressed. So you can go between the spatial map and this lineage tree. So that's how one example of how they do spatial transcription. Another way is to actually get some of these examples here. These are different next-gen sequencing techniques and some other kinds of techniques that they're doing here. This is spatial transcriptomics. So this is a database for what they call spatially resolved transcriptomes. So this is like different methods that they're using for this. This is basically where they're able to resolve transcription to different tissues, different places in the organism, and they have different methods. So there's spatial transcriptomics is but one of these. Let's go to this tab. And so this is, again, I think a data set. So this is the data set that they're presenting here. Let's see, by positioning histological sections, which are sections of a tissue, on arrayed reverse transcription primers with unique positional barcodes. It's just the method they use. This study demonstrated high-quality RNA sequencing data with maintained two-dimensional position information for the mouse brain and human breast cancer. So they're able to look at like different sources of tissues. They used an adult mouse, and this is kind of a model. This is a strain of mouse here, so don't worry about that. And it was less than 2 months old, so it was a juvenile mouse. And then they looked at the olfactory bulb within that mouse. And they also used a breast cancer biopsy, I guess from an adult human of some person, I don't know. They didn't give any more detail than that. So it's human and mouse, and they're looking at a subset of genes in the mouse and the human, and then, so that's how they do this. And so this is an example of the mouse olfactory bulb. So this is a juvenile mouse olfactory bulb. You can see that they have this is a section of the tissue, so they've cut through the tissue at a certain orientation, and they've taken a slice of it. And then they prepare the slice, and they do this analysis. And so you can see that there are these different spots on this slice. And this is a sample of, what position this is, what the expression level is. So you can see X and Y there. It tells you the X and Y position on the slice, and then the expression level. So what you can do with this is actually kind of interesting. You can run something they call spatial statistics on it, which is where you take a bunch of points and you figure out the distance from, say, like some focal point. And you can find trends in the data. You can find like the significance of like spatial distributions using these spatial statistics techniques. And I'm not going to get into those here, but those are some techniques that people often aren't familiar with because they're not used everywhere. They're usually used in like geography and in earth science, but those methods exist. Another thing you can do is, of course, signal processing or some other type of frequency analysis, and so that's something that is also useful. It tells you different things about like the periodicity of some of these expression patterns and other things. So this is an example of a mouse olfactory bulb, like a slice through it. And this shows you what you can do with this kind of data. And this is an intensity map, so this is normalized expression level. Each of these points show, like, you can see the color. So at the edges, roughly, of like this bulb is split into two parts. At the edge of each bulb, you have this high intensity expression. In the middle, you have this low intensity expression. But to express that in statistics is a different story. Then there's this paper that I found on using Polyarray Maps for analyzing complex hierarchies in single-cell data. So this is a paper on, this is kind of using what they call scRNA-seq data. So this is kind of refine some of these methods for analyzing these samples. So existing techniques are based on Euclidean geometry, a suboptimal choice for modeling complex cell trajectories with multiple branches. To overcome this representational problem, we propose Polyarray Maps, a method that harnesses the power of hyperbolic geometry into the realm of single-cell data analysis. So hyperbolic geometry is another way to project things. Euclidean geometry is where we have that XY map. So it was a flat map. It had a flat surface. There was no reference for curvature at all. But this actually gives you the ability to incorporate curvature in your model. And so often understood as a continuous extension of trees, hyperbolic geometry enables the embedding of complex hierarchical data in only 2 dimensions while preserving the pairwise distances between the points in the hierarchy. So it allows you to look at like curvatures and preserve the distance between points, because when you have a curvature, sometimes the points don't translate. You can have distance between 2 xy points, and there's a Euclidean distance, and that's easy to understand. But when there's curvature in your sample, in your surface, a lot of biological systems, if it's not a flat slice, it's going to have that curvature. This is a nice way to represent that curvature. this enables the use of our embeddings in a wide variety of downstream data analysis tasks. So they're doing a lot of things afterwards that are going to be, this is going to be useful in. So this is a single type of embedding. So we talk about embeddings in graph neural networks. This is another type of embedding where you have this reference frame, this reference sort of space, and then you map your data set to it. And then Polyarray maps are a certain subset of this type of hyperbolic map or hyperbolic geometry, where you can produce state-of-the-art two-dimensional representations of cell trajectories. So let me go to see if there are any images here that we can really dig into here. This is a good example here of how Polyarray maps discover hierarchies and branching processes. So this is what they call Waddington's landscape or epigenetic landscape. This is a single cell or a single thing in development, and it's going through these transition points where you start off with a single cell, and it divides into 2 cells, and then those 2 cells divide into 4 cells, and they take this trajectory through developmental space. So you end up with 4 cells that have sort of different developmental trajectories from a single cell. And so this is just kind of what they're getting at here. These trajectories then can be mapped from this conceptual landscape to a hyperbolic space, which is actually a metric space that you can measure distances on, or measure distances between like expression patterns, because these channels down this landscape are only hypothetical. They actually represent changes in sort of the expression patterns of the cells and so forth. So it's kind of a very conceptual thing. This gives you you're able to put some numbers on it and able to put the cells in some sort of reference frame that you can compare and contrast. This is an example of Euclidean space where you have these pairwise distances between these cells. And it shows that, you have this space that's linear. It's not going to preserve distances for multiple, like for spatial relationships and distances between, say, like if you have a cell comparison map and you have differences in expression and you have a distance metric of difference versus a spatial distance metric, those two things don't map well to one another. But you can preserve these pairwise distances by using a Polyarray map. And so it's a circle here, and you have these cells that are at these locations in the circle. And so this is the way they do this. I know there's another image in here that shows you kind of what this looks like for different samples. So this is where they have an embedding quality metric that they use. And so this is an example of sort of fit, I guess. And so for different species, different samples, they use these different embeddings. Polyarray maps are in blue versus some of the other ones that they use. In genomics, they use a lot of these type of embeddings to represent the data, and they just try to find the best one. So they have UMAP, TSNE. These are dimensionality reduction techniques you're probably familiar with. PCA, of course, which is principal component analysis, and some other ones that they use that I think are more distinct with respect to genomics. But they basically are comparing these, and so you see the Polyarray maps usually consistently end up on top. So they end up at the top, this good end of the y-axis. So that's good. And then this is the image I wanted to show here. So this is an analysis of the C. elegans cell atlas. This is where we have our cells from C. elegans that we saw in some of the previous papers. We see that we have some things that we can identify, different cell tissue types of body wall muscle, choliomyocyte, germline, Z1 through Z4, which are, I believe, germ cells, ciliated amphid neuron, ciliated non-amphid neuron. So you have all these different cell or tissue types that are put onto this embedding. and they have these distances between each other. So you can see that there's a distance between ciliated non-amphid neuron and body wall muscle, and you can see that the distance is here. This is, I guess, an expression data, so this is a little bit different than the type of thing we're looking at with lineage trees. And it's a different way of thinking about it because, if you are familiar with T-SNE and UMAP, you know that those are very hard to interpret. I think we've talked about this before in meetings where you have this multi-dimensional data set and you boil it down to like 2 or 3 dimensions, and then you build this map, and it looks really pretty. This actually kind of looks like a UMAP representation, but you can't really interpret it. And so it is with this method as well. It's kind of hard to interpret, but we can see, we know what the distances are, but beyond that, we don't really have any good interpretive theory of why these things are, far apart or close together, except to say that those things cluster together in this analysis. So we know that they're similar. Other than that, we don't really know that much. And so this shows you, again, with embryo age, where these cells are coming up and what they are. So here's the germline, and then you see other cells coming up later in development to 650 minutes. And so these cells from this map are, differentiated in time, a germline cells being first and then going on to these cells, which are the last ones to differentiate. And so you can look at cell type, pseudotimes, age of the embryo, and represent all of these in the same embedding. So this is, again, pseudotime, I guess, is some sort of like time measurement. It's not like minutes, but it's maybe order. And so, one can do these kind of maps for different types of, different criterion. So cell types, the cell, if we classify the cell, C. elegans, we know if we have a lineage tree, we know what the adult cell is going to be. So we can actually classify things in development as the adult cell. Pseudotime is like when these things appear, and then the age of the embryo is like the age of the whole embryo versus when that cell appears. And so that you can do a lot of things like that and build these kind of maps. Again, they're very beautiful, and they're somewhat informative, although not completely informative. And so that's kind of the idea there. Okay, Ishan had to go. Thank you. So, yeah, any questions about that? I know that was a lot. I know Karan had started this conversation about adding in transcriptomic data.
Yeah. Especially gene expression related data. Even the previous paper, I think, had a lot of insight related to possible features that could be added. And I'm thinking maybe, how about adding another, like a bit more general model, for even C. elegans. I don't know if that will be useful for C. elegans as well. I think I'll have to look more into it. But I think it could solve both of these issues.
Yeah.
And start with the C. elegans model and then see if I can adapt it to the axolotl thing.
Yeah, C. elegans.
I have a question completely off topic.
Okay.
Do you know what the mechanical properties of the nucleus are in most cells? Is it softer than the surrounding cytoplasm, or is it harder or more elastic, or do you have any clues as to that?
Well, I don't know if there are any studies that have characterized that. I think because there's a lot of DNA in the nucleus, it might be harder, but I don't know because there's a lot of stuff in the cytoplasm too floating around. So it's hard to know. I mean, the cytoplasm is likely to be anisotropic. There's cytoplasm, which is the fluid, but it's like there's stuff in there. And so any measurement is going to have to take that into account, yeah.
Yeah, it's packed.
Yeah.
It might match it, kind of. I was thinking because it's all one cell, it probably has some sort of matching. Like its elasticity will probably match the elasticity of the cell. Anyways, you don't know of any studies on that? No?
Well, I'm not familiar. I mean, there might be some, biochemistry, like in the biochemistry literature, there might be something, because I know that sounds like a very biochemistry oriented question. Um, like, what is the specific density of the? It's like in a textbook, you see like you have the cytoplasm and it looks almost empty, which is wrong. But and then the nucleus, which is this shell, and it has like the DNA in it, but there's a lot of space in there. And then the cytoplasm is a liquid, but it's like, there's it's I guess a thick density. I don't know what the viscosity is of it, but like you have a lot of things in there, so it's like going to be very hard to. I mean, I would imagine it would be, yeah. Imagine it would be highly anisotropic, and it would be, actually kind of hard to measure because you have to get a single cell, and you have to get isolate the nucleus, and
and then squash it. Yeah, people have done that with optical tweezers. Maybe I should. Yeah, maybe I have something.
Okay, yeah.
Okay.
Yeah, optical
Right,
tweezers
thanks.
might be it. Yeah. So back to Karan's point, I think like for C. elegans, that's a good system to work in because there's not only it's easy to work with in terms of lineage tree, but there's also a lot of data available, like secondary data. And then people have done studies like the one I showed you, the first one I showed you, where they did, they were able to do everything in one shot.
Okay.
So, I mean, and then putting together data for like C. elegans, it's actually easy because you have it's easy to align it because there isn't a lot of variation between experiments. You have each cell has an identity and it comes up at about the same time. And so that's a place maybe where you can do a lot of that sort of thing, like combining the data sets and things. If you're trying to do that, and it would be much harder to do it in axolotl because we have there's variability in how the cells are differentiating in time, just enough that it would be hard to do that to build something that was really consistent. So, yeah.
Okay.
Yeah. So any other questions at this point? Okay. I'm going to go over some papers in a bit, but I wanted to first go over we have the major task board, and I bring this out every so often just to kind of give an idea of where we are on it. We have a lot of action items that we need to follow up on. I think some of these have been done. I think this one is done. This one is, sometimes we come up with things in the meeting and then they drop. And then I come up with something else. And then, sometimes they get addressed, sometimes they don't. So this is actually on our DevoWorm GitHub. It's group meetings, projects. So this is where we keep a lot of this accounting. We have things like the 1D Ising model. I don't know if that ever went anywhere. I think people are still kind of thinking about it. We have this hypergraphs submission was something that I showed last week, and the submission is finished, but now there's some more work to do on that. Apply to GSoC as our own organization. So unfortunately, we didn't get selected as an organization for that, but we're still participating through INCF. We actually applied as our own organization this year, but that didn't happen. That's fine. We're just trying to maybe get more slots. Some of these things are really old. I'm going to go over this later, but I know that Jesse's sometimes works on this. I work on this sometimes, so just so people know, there are a lot of kind of open issues. So if you're looking to contribute to the community here, we have a lot of open issues, a lot of things to follow up on. Just mention some issue, by number, and if you're interested, and I can kind of give you an idea of where we are on it. That's the best way to kind of keep track of all these moving parts that we've had. So now I'm going to go to papers here. And so let's see, what do we want to talk about this week? Let's
Um,
see.
Could
Yeah,
I,
question.
before you start, I'm participating in the APS physics March meeting,
Okay.
and there are quite a few people who do networks of various sorts in that meeting, but there's soft matter physics in the meeting all over the place, with cytoskeleton, whole sections on cytoskeleton.
Okay.
So it's physics, but it's related to biology. They have quite a few sessions. Just thought I would mention it.
Yeah, that would be great. And then we have that special issue that we're working on. Maybe we could find some people there who might want to contribute to that.
Yeah.
Yeah.
I'll do a bit of research and get you some names.
Yeah, because it's kind of like just identifying people might be likely to contribute. That's the key. Yeah, so that sounds like an interesting session.
Okay. Yeah, it is so far.
Oh, okay, yeah. Well, Gopinath, welcome. Okay. So let me go back to this. What is this one? Okay. Yeah, why don't we talk about this? So this is kind of we call life as emergent phenomena. And this is about how life is sort of, we talk about like how things are emergent when we talk about whole organisms, as opposed to like gene expression. We're talking about like biophysics measurements of cells. We talk about the embryo.
I can't see. Sorry, I can't see your screen.
Oh. Let me see. Maybe I'm not sharing my screen. No, I'm not. Okay. There we go. Okay. So when we saw that embryo with all the cells and how the cells are dividing and forming shapes, this is something that people have argued is what they call an emergent phenomenon. So this is where you have things that interact, parts that interact, sometimes a lot of parts that interact simultaneously, and they form patterns or they form things that you can't really explain by looking at like simple pairwise you look at pairwise interactions, but it's not simple causality, or it's not some simple rule that you apply universally. There's this emergent phenomena that happens where things sort of spontaneously organize, and it sounds like magic, but people have debated this for many years, and so people have tried to study in different ways, and it's kind of hard to study because how do you study a bunch of interactions simultaneously, and how do you get to the structure of those things? So sometimes people use tools like network theory, which is something we've talked about. People use different types of scaling laws to explain this is how the interactions occur in time or in space, and so those are ways you can explain it. In this paper, they talk about life as an emergent phenomena, studies from a large-scale Boyd simulation and web data. So in our group, we talk a lot about maybe the emergence of life, or the emergence of embryos, or the emergence of biochemicals. And so, there are different rules there, but a lot of people think that emergent phenomena have rules that transcend any one system. So if you look at multiple systems and compare them, you'll see commonalities in them. And what they do in this paper is they have a large-scale Boyd simulation and web data. So they're using two separate things, and they have nothing to do with biology, but this is something that, is also classified as emergent phenomena. So this is a Japanese group. And so a large group with a special structure can become the mother of emergence. We discussed this hypothesis in relation to large-scale Boyd simulations and web data. In the Boyd swarm simulations, the nucleation, organization, and collapse dynamics were found to be more diverse in larger flocks than in smaller flocks. When they talk about Boids, they mean this type of simulation, and I don't know if people are familiar with this. The computer science people might be familiar with this, but it's a simulation of basically bird flocks. And they did this back in the 80s where they had these sort of digital birds that each bird has a set of rules on board that sort of govern their interactions. So it's like each bird will have a set of rules that say, don't get too close to your neighbor, don't get too far away from your neighbor. When your neighbor turns, turn with the neighbor. And it's basically like, well, we talked about cellular automata, where you have neighbors and the state of the individual depends on the state of the neighbors. So they basically use a rule, they calculate a rule based on the state of their neighbors. Now, if you do this in parallel, if you find that, you can have these kind of cascading effects where the neighbor influences another neighbor, and you get these kind of patterns where there's collective behavior, there's collective movement of a bunch of agents, there's activation across a grid that is, organized and looks like something that's like a blob that's moving, and there are a lot of lifelike aspects to it. So that's what they mean when they say Boyd swarm simulations. They're like bird swarms, but they're really simplified bird swarms. They're actually implemented on a computer. So they found that in these Boyd swarms, a lot of these dynamics, they say nucleation, organization, and collapse, they mean the patterns of the swarm. So a swarm is a collective of agents moving around in a coordinated fashion. Those swarms can collapse, they can nucleate into smaller swarms. Those are more diverse in larger flocks. So if you have more Boids, like say 1,000 Boids versus 100 Boids, there's a more diverse set of phenomena that you can look at. As you may guess, they're not highly predictive. In other words, if I do one simulation, I observe one type of, say, flocking versus another simulation, I see another type of flocking. It's very highly variable, but they have basically the same behavior. Those behaviors are more diverse, but they still resemble that same flocking behavior. And then the more agents you have in a simulation, the more diverse that is. In smaller flocks, like maybe 100 agents, their behavior is much more uniform across runs, simply because there aren't a lot of options. I mean, if you think about what I just described, there aren't a lot of options. I mean, there are only maybe 100 Boids, so they don't have a lot of divergence across the flock. It's pretty consistent, but in larger flocks, you have a lot more distance, say, between one end of the flock and the other. So there's a lot of opportunity for noise to enter the equation, or if the flock goes around a barrier, there's an opportunity for bifurcation. There are a lot of things that happen at larger numbers in terms of noise, in terms of variability. In the second analysis, which is large web data, which I guess is like looking at this network of sites on the web, consisting of shared photos with descriptive tags, tended to group together users with similar tendencies. So they're using a database here of shared photos with descriptive tags. They're looking at users and what their photos are and what the descriptive tags are. And so they're actually looking at how that forms a network. And so this is network science. It's a little bit different than flocks. This is where you have a network of users connected by these common photos and common tags. And so this type of thing, you just basically have a bunch of users. You look at how they're using photos and putting together tags, and then you see how that network grows over time. So this network is able to develop a core periphery structure. So a core periphery structure is actually something that is sort of a signature of hierarchy. So if you have a core and periphery, what a core and a periphery are. This is a core, this is a periphery. In a network, sometimes you'll have like a node here, that it connects to a lot of other nodes. Sometimes you'll hear about this in terms of a rich club network. Sometimes you'll hear about this in terms of a hierarchical network, or something like that, where you have this node that's in the core and then the periphery nodes are out here. This node has a high number of connections as compared to these nodes out in the periphery. So this is the idea. You have maybe a core node that maybe organizes a lot of things, and then the periphery, which are the nodes that are less connected, that are less important to the network. Because this core then is connected to other cores. There might be another core out here, and so forth. And so that core is an important point in the network for traffic to flow. If you think about an airline network where you have hubs, like Chicago or Los Angeles or New York in the US, those are cores of the airline network, as opposed to periphery places that aren't connected to everywhere else. So this is the core periphery structure of a network. We show that the generation rate of novel tags and their usage frequencies are high in the higher order cliques, which is a measurement of areas of the network that are very highly connected to one another. In this case, novelty is not considered to arise randomly. Rather, it is generated as a result of the large and structured network. So again, we get this theme of having to have a larger structure in place to get a lot of variation. So this novelty and variation is generated by having a lot of nodes or connections or voids or whatever. We contextualize these results in terms of adjacent possible theory. So this is an interesting thing. So adjacent possible theory involves having this. So it's sort of like the core periphery idea, but instead of having a core and periphery, you have this grid. Let's say we have a huge grid here of 1,000 cells, and we're in this cell here. Okay, now let's suppose that there's a path. There are a number of potential paths we can take from this point, and they're all grid points. So you can go in this direction, you can go in this direction, in this direction, in this direction. So now it's a matter of figuring out where you want to go, and then when you go to that place, what are the adjacent grids or grid cells that you can go to? So let's say you can go to these two places. Okay, and then if you're here, after the third step, where can you go from there? And it turns out you can go 3 places, right? So now look at what we've done. We've taken a trip from this cell to this cell to this cell to this cell. Okay, so in 4 steps we've gone quite a ways across our graph or our grid. And the idea is that each step you take unlocks a new set of possibilities. So the adjacent possible means it's an adjacent state, and then that opens up another adjacent state. And if you take a path through those adjacent states, you end up with new possibilities, and you end up in new places in the grid or in the world. And so you can end up very far away from where you started based on just kind of navigating through a structure. And then it depends on what's available to you. So, you think of this as configurational entropy, right? You go to one cell and then the next cell and the next cell. And when you get to that maybe 4th cells out, you are in a different part of the grid, but you also have different possibilities in front of you. But if you had taken another path, you might take 4 steps out. You might have a whole new different set of possibilities open to you. So if you go out this way, there are new possibilities out here and so forth, and you end up in a totally different place. And so that's what they mean by adjacent possible theory. They're proposing that that's a new way to understand collective intelligence. I think that's an interesting point, because if you have that kind of a system where you have an initial condition or an initial point, and then you start moving across the grid and you get to these new places and you have new possibilities, and you end up after maybe 4 or 5 iterations in totally different places. This is something that I think they stress a lot in complexity theory, that you have an initial condition and your system evolves a way to different novel states. It's a similar thing here, where you have different possibilities in front of you. You end up in totally different places. I think that's an interesting way to see how these systems navigate complexity. That emergent systems aren't like necessarily predictable. They're predictable in the sense that they end up anywhere in this set of possible states, which could be, hundreds of thousands of states. But beyond that, we can't really predict where they'll end up. And so this goes back to a lot of complexity theory and a lot of this sort of these sort of contingencies. And that's actually can link us back to development, because development thrives on contingencies. When we have a single cell, the cells divide, and then those cells divide to their own daughter cells, but that constricts, say, where they can be in space or what their fate can be. So there are a lot of constraints on what that next set of states can be, but it takes you in very different directions. So like one cell lineage in a development might evolve down this path, another cell lineage might evolve down this path, and you get two very different sublineages, but they all originate from the same mother cell. And so that's one way to look at emergence. And so this paper kind of goes, there's a special issue that this is associated with reconceptualizing the origins of life that might be of interest to people. This is a Royal Society paper. So they kind of go through a lot of this. Oh, they show the Boyd simulations here. So these are the Boyd simulations, swarming behaviors. So you see you have your agents in a space, and then they have this, so as you add agents and they move around, they start to swarm and flock in these aggregates. And you can see the very complex patterns that they take, the shapes that they take on. You see thicker groups here because there are more agents, and you can see what they look like. They're very highly nonlinear, very highly, they almost look like turbulence in a flow. But that's kind of the idea too. In turbulence, you see some of the similar things that are going on here. Okay, so then the second paper in this series is a blog post by someone called, his name is Johan John, and he does a lot of philosophy, and actually he's a neuroscientist, I think, by training. But he has this nice blog where he talks about a lot of ideas that are really kind of, theoretical. And this post is called "What is Emergence and Why Should We Care About It?" So here he shows some shapes that have sort of these emergent structures in them, and how you can basically use the same assembly rules to get different types of shapes. And he says emergence occurs when there is a conceptual discontinuity between two descriptions targeting the same phenomena. This does not mean that emergence is a purely subjective phenomenon. So like I talked about with the adjacent possible, you could have two totally separate outcomes from the same sort of map. Only that the scientific double coverage may be a good place to look for emergent phenomena. So he's, for example, in the case of starling murmuration, which is a collective behavior that they do, there is an aggregate description of individual birds and a description of the flock and its unified entity. So you can describe individual birds and what they're doing, and you can describe the state of the flock, because you saw the flock can be like, have the shape, it can look like a comma, it can move around like a blob, it can have this sort, it looks kind of like turbulence. And you can also describe what the individual birds are doing. You can use like a set of rules to describe their interactions with other birds. But you can't necessarily get into sort of that middle scale, which is, what you have in different parts of the flock, what are going on there, or that collective structure. And so this description of the flock as a unified entity invites descriptions in terms of concepts from fluid dynamics. So this is something that, we talked about in fluid dynamics with respect to flows and turbulence. And so turbulence actually has a number of parameters that you can use to describe the flow. And it's, but it depends on you describing the entire flow. It doesn't depend on you describing the individual particles that are in the flow and forming the flow. So this is the sort of thing that we don't actually have. Descriptions of individual birds or particles that are, and then linking it to the larger collective, to the larger scale. So that's something we don't have. We can only describe that individual particle or bird in terms of what it's doing individually, not as what it's doing collectively. And then he gets into this idea of phase transitions in physics, which is where you look at changes over time. So those flocks can transition from one state to another. A flow can transition from one state to another. And in phase transitions, you have different phases of matter. So you can take matter that's a gas and it can transition into a liquid. And it does this fairly quickly. And so what they call that is a phase transition. It's a quick change of state. So these phase transitions do not fully map onto descriptions of other phases. So if I have a gas and it transitions into a liquid, I can't really describe that liquid as a gas anymore. I have to describe it using a different way a different set of criterion. So surface tension, for example, is not defined for gases, since gases do not have surfaces. But when you have that phase transition to liquid, suddenly surface tension can be measured. In the transition from gas to liquid, a qualitatively new attribute not only emerges, it becomes a defining feature of the post-transition system. So this is, again, when you have these phase transitions, it brings new properties into being. And so it kind of goes through a lot of this emergence and ontology, and, kind of talking about how emergence is controversial. And a lot of that has to do with the language that we use and the descriptors that we use. So that's an important thing to think about. And then, of course, finally, this idea of reductionism. So we're used to reducing systems, physical systems and biological systems, to their smallest physical parts or their smallest constituent parts. And with respect to emergence, you can't do that. You can use a reductionist approach to look at the individuals, but it doesn't really do much to describe this collective. And so that's the idea that we're trying to get here, is that we're definitely not talking about a reductionist system. And although we can reduce these systems down to specific individual constituent parts, it doesn't really do much to describe what we're looking at. So the parts of one discipline, say cells in the context of organ-level biology, are the wholes of another discipline, cells in the context of molecular biology. So we have, parts can be wholes, and so that's another thing to consider, depending on your perspective. If you're a molecular biologist, the cell is a whole. If you're an organismal biologist, the cells are parts of a larger whole. So your perspective changes your interest in what's emerging. So, like, you might be interested in the emergence of, maybe proteins. And as a cell biologist, that's something that's emergent to you. But you don't see the cell as being part of an emergent system. But an organismal level biologist would see the cell as being part of an emergent system. So that's all I'm going to talk about on that. I think that was a good overview of that. And I thought that was valuable considering some of the things we've talked about here. Gopinath couldn't get his microphone to work. Yeah, sometimes it doesn't happen, but okay, he'll send me an email later to introduce himself. Well, thank you. And I assume you're here for GSoC. So we record our meetings, and I can send you a recording of this so that you can get some of the information I talked about earlier in the meeting, which was how to write a proposal. So that's good. And then Harikrishna had to leave. So thank you for attending, Harikrishna. I also had, I think, let me see, are there any other messages in here that we had one more? Okay, yes, for GSoC, good. Okay, so, so thanks for attending the meeting. We have any questions before we end the meeting? Or?
No, I'll maybe report back to you on some of the interesting things in APS physics.
Okay, yeah, it sounds good. Yeah, look forward to it. All right. Have a good week, everyone.